Welcome back to the VJHemOnc podcast, where we bring you the latest developments and expert perspectives from the world of hematology and hematological oncology. In recent years, targeting mutant calreticulin, or CALR, has generated excitement in the field of MPNs. Last month, we had the pleasure of attending the 30th Congress of the European Hematology Association, where the late-breaking abstract presentation of a trial investigating Icahn 33989, a mutant CALR-targeting monoclonal antibody in essential thrombocythemia, sparked expert discussion. In today's episode, you will hear the promising results from this study, as well as additional expert insights into the targeting of mutant CALR in MPNs. First, Lucia Masarova provides insight into what makes mutant CALR an attractive therapeutic target in myeloproliferative neoplasms.
It's just a different driver of the disease where calreticulin, again, binds on the surface. So a JAK2 mutation remains inside the cells and MPL works for the thromboreceptor. So calreticulin, it's probably the first novel exciting discovery where these mutant calreticulins travel on the cell surface, and that's why they're open to immunologic address and approach. So it's easier to develop antibodies and then hopefully with that to attack them and basically attach to calreticulin mutation and then get rid of them. That's a similar principle we have across hematology or even solid tumors, where we know that cancer cells express certain mutant proteins or different markers, where we are able to develop drugs. Either it's a simple antibody, or it's antibody that's armored, or it's an antibody that engages the immune system, or even the chimeric antibodies that we have approval across hematology and almost every single cancer, but nothing in MPNs. So this is a first opportunity, first in human and proof of concept approach, where we will be able to go in the target, which is specific for the cancer. Mutations in calreticulins, who these people are mutants, so they're only cancer cells.
Next, Jean-Jacques Kiladjian outlines CALR targeting approaches that are being explored in clinical trials and discusses the disease-modifying potential of these therapies.
Targeting mutant CALR by antibodies, with antibodies, is really something we wanted to see for many years. And hopefully we have now at least 2 clinical studies that have started for a while, dose escalation studies. One of them, the first results will be presented for the first time during this meeting at EHA concerning an antibody, which is a neutral antibody that targets mutant CALR developed by the Incyte company. And we will show the results on the mutant CALR allele burden over time. So these are the first preliminary results showing really spectacular molecular responses with a huge decrease in the mutant CALR allele burden on treatment with this antibody. There's another trial with a B-specific antibody, which is a different strategy, but both are addressing the same question of trying to get rid of the mutant cells and hopefully change the natural history of the disease. So preliminary results, but very encouraging data on molecular responses, at least with the first of these antibodies. We have big expectations with these new strategies targeting the clone itself with specific drugs and not only JAK inhibition. That has very important issues that are mostly to address not only the mutant clone, but also sometimes inflammation, for example, for some of the JAK inhibitors and other targets. But here we are just targeting the malignant cell itself, the source of the cell, and hopefully the leukemia initiating cells. So we hope that with this type of drugs, we may eliminate the mutant clone, the malignant clone, and hopefully maybe achieve a cure for some of these patients. That is not the case currently with JAK inhibitors.
Now, you will hear John Mascarenhas share the results of the phase 1 trial of Icahn 33989 in patients with ET, which he presented in the late-breaking abstract session at the meeting. Following this, Claire Harrison provides further insight into the study, as well as mentioning other targeted therapies being explored in MPNs.
Today for the late-breaking abstract, we presented the initial results of 989, which is a monoclonal antibody to mutant CALR TPO receptor complex. This is a treatment in development that is supported by preclinical modeling, demonstrating selective targeting of mutant CALR hematopoietic stem cells and the ability to deplete that pool and potentially change the course of disease. So the preclinical data look very good. We took it into the clinic in a dose escalation phase 1 study, starting at 24 milligrams infused every 2 weeks, all the way up to 2,500 milligrams. This was really a very large global effort. So there was really 2 studies, one in the US and then one outside the US, combined data set demonstrating, most importantly, safety, very well tolerated drug, very few events that were significant that were attributed to the antibody. There was one thrombotic event, splenic vein thrombosis, when the patient was treated at a low dose in a patient with risk factors for thrombosis, including a history of thrombosis, so unlikely related to the drug. So patients tolerated the drug very well. We saw lipase increase that was mostly grade 1 and 2, unexplained, with no clinical or radiographic consequences. So we were very happy with the safety and tolerability. In fact, we did not have a DLT, nor did we achieve a maximally tolerated dose. When looking at efficacy, we saw very rapid and durable reductions in normalization of the platelet count without thrombocytopenia. And then we looked at the biomarkers of disease modification, including reduction in JAK2 V6-1, reduction in mutant CALR VAF. And we did this through single-cell RNA sequencing, demonstrating that by immunophenotyping in the hematopoietic stem cell and myeloid erythroid lineage cells, significant reduction in mutant VAF was seen. And this was also correlated with clinical responses. Additionally, in the bone marrow, we saw a reduction in mutant CALR staining megakaryocytes, with an increase in normal megakaryocytes, suggesting shifting to normal hematopoiesis. So in 2 compartments, biomarker evidence of disease modification correlating with clinical activity, reduction and normalization of platelet counts. I would say this is probably one of the most exciting abstracts, I'm biased, that I've seen recently, because it's rare to have a phase 1 study with such well-tolerated and early clinical activity, both at a clinical and biomarker level. It's paramount we continue to follow these patients out. It's paramount that we expand probably several dose levels to really look at efficacy and ultimately create a clinical development pathway forward for mutant CALR ET. At the same time, we're also evaluating patients in these studies for myelofibrosis, mutant calreticulin myelofibrosis. That data set will likely be shown at upcoming meetings too. So very exciting times for immune therapy, which is, I would say, novel for this disease type.
So I think this is such an exciting time in MPN at the moment, because we have the opportunity to see the first data now coming from the mutant CALR-targeted antibody. And I've personally been so excited to be able to offer patients that treatment and to see the results presented by John Mascarenhas at EHA in the late-breaking abstract session. And what we presented in that session was data from over 40 patients with ET treated with this antibody, actually in 2 studies, because the dosing in North America was different from the rest of the world. And the rest of the world, we went much faster and treated patients up to 2,500 milligrams, whereas the increments in North America were slower, as mandated by the FDA. What was very striking from this data is, one, that platelet control happened very quickly within 2 cycles. So what the patients receive is an infusion that lasts an hour. They have it every 2 weeks. And the platelets were generally normalizing within 2 cycles. These are patients who failed at least one therapy. They were allowed concomitant either anagrelide or hydroxycarbamide, but for most patients, they had stopped that, including my own patient. The infusion is very well tolerated with, very, very few grade 3 or above adverse events. Lipase increase, which we don't really understand, but wasn't associated with any, pancreatitis or liver abnormalities, etc. And we need to dig a bit more into why that actually happens. But we do see sometimes lipase abnormalities, for example, with JAK inhibitor therapy. And also beautiful data. So if the listeners have the chance to watch that online, what we saw was also rapid reduction in the CALR VAF. So clone size reducing, but also mutant megakaryocytes, beautifully stained bone marrow showing mutant CALR on the surface. And then after 24 weeks that these mutated megakaryocytes were reduced, but also really importantly that normal megakaryocytes were increasing. And we also showed some data with regard to that as well in terms of progenitors. So clinical efficacy well tolerated, and then the beautiful biological data coming along underneath. I think it's so important and really great to see. There'll be more data presented doubtless at ASH. There are MF patients also being treated with that. There are MF patients being treated with the Janssen T-cell engager antibody. Hopefully we'll see some data with regard to that. They also treated ET patients. And then there was, just sticking to the topic of CALR, of course, there's the CAR T-cell developed by Alex Rampotas from the UK and presented at EHA by Beth Sala, a T-cell engaging CALR specific antibody from Insight. So these agents will all be really important to use for patients, varying potency, of course, and it's important to remember that we know that patients, especially with myelofibrosis, don't have very intact immune systems. So we may well need something a little bit more potent and something that can penetrate the fibrotic marrow. And then a word on other targeted therapies, such as those targeting the V617F mutated JAK2 and the pseudokinase domain of JAK2. Also, we should expect to see some data. We haven't seen any clinical data with these agents yet. They're oral agents and potentially could offer a lot of benefits for patients, especially avoiding off-target harms potentially, such as skin cancer, which we do unfortunately see with current JAK inhibitors, but also perhaps offering more potent effects on the clone. So super exciting. Looking forward to discussing in more detail in our post-EHA webinar, but also seeing the data unfurling later on.
As mentioned by Professor Harrison, CAR T-cell therapy is being investigated as a potential approach for targeting CALR in MPNs. To conclude this podcast episode, you will hear as Alex Rampotas and Zoe Wong provide insight into their work, which focuses on the development and evaluation of the first-in-class CAR T against CALR mutant neoplasms.
So in myelofibrosis, we had a few treatments that became available very recently, but none of these treatments have had a curative effect. None of these treatments have been disease-modifying. And the only treatment option available for patients is an allogeneic stem cell transplant. However, this treatment is only available for a small number of patients. And even in those cases, up to 25% of patients, they would have seasonal-related mortality. So it's a very toxic treatment. So we do need better treatments and targeted therapies. And mutated calreticulin is a fascinating molecule because the mutation enables the mutated protein to escape the endoplasmic reticulum, to bind to the thrombopoietin receptor, and to be expressed on the surface. And this leads to aberrant activation of megakaryocytes and stem cell and drive the disease. The other important thing is that the mutated protein is very unique because the C terminus is novel, and that means that we can develop specific immunotherapies against it. And there have been a few immunotherapies recently. There is a bispecific T-cell engager and a blocking antibody against it. But during our project, we developed a novel second-generation CAR T-cell therapy because CAR T-cell immunotherapies is the strongest immunotherapy that we can develop. And CAR T-cells are able to have direct cytotoxicity. They can bypass the need for MHC antigen-dependent presentation. So potentially, they are able to overcome some of the immune suppression of myelofibrosis and directly eliminate the malignant stem cells. So during the first phase of the project, we developed this CAR T-cell and we tested it against cell lines that they were carrying the mutated calreticulin target. And we have identified excellent results. The CAR T was able to eliminate the cell lines specifically, and it was able to do that on in vitro and in vivo in the NSG murine xenograft model. And also we have identified excellent proliferation of the CAR T cells. They maintain a quite naive phenotype after multiple rounds of stimulation. And overall, we're very quite happy. But on the next stage of the project, we want to test this CAR T cell therapy against primary cells, CD34 positive cells, isolated from actual patients that they have myelofibrosis. And this is when we formed collaboration with the Cellular lab in Oxford, and Zoe Wong performed some of this work.
Yeah, so as Alex said, we have taken this first-in-class novel CAR T and tested against primary patient cells. And we've tested this CAR T against cells from patients with type 1, type 2, and type 1 and type 2-like mutation. and seen great results. We've seen highly specific cytotoxicity with the CAR-T, with very limited off-target effects. The other exciting thing that we've been able to do in this project is look at the efficacy of the CAR-T in a relevant human tissue microenvironment. A major problem for preclinical validation of immunotherapies is the fact that a lot of our preclinical testing is done in a murine model, which is non-specific, and also done in a dish. So with our human bone marrow organoid model, what we've been able to do is test the efficacy of the CAR-Ts against primary patient cells in a 3D microenvironment, or a 3D rendering or model of what the actual patient's myelofibrotic niche would be like. And we've been really excited to dive into how CAR-Ts and hopefully other immunotherapies like BiTEs or inhibitory antibodies as well, and to form in a niche or a microenvironment that is fibrotic or immunoregulatory, which is a major problem for patients as well.
So what do you think are the advantages of this organoid model in comparison to using mice?
The organoid model is scalable, so we can make many organoids and test different doses of CAR-Ts, different timing, synergy with other existing therapies, and that would be very limited in a mouse model. And also before moving on to patient or clinical studies, we can kind of refine what types of dosing and timing we want to move forward. So that's a huge benefit. Another benefit in comparison to mice is that in a lot of the mouse models, we actually have difficulty setting up models that faithfully represent the human microenvironment and models that are immunocompetent as well. So this model allows us to really carefully think about what elements of patient microenvironment we want to recapitulate, and we can test efficacy of immunotherapies with and without certain immunoregulatory factors as well.
I think that's making a very good point, because if we were to test, for example, our CAR-T cell in a murine immunocompetent model, we would have to use murine CAR-T cells. And although it would have been another complex model, actually the CAR-T cells would have been different from the final product that might go into humans. So I definitely agree that organoids may be the future of drug development because of all this versatile ability to test all of these different things and to recapitulate the actual human bone marrow rather than testing drugs in a murine environment.
Yeah, we can also look at off-target effects and see how the different therapies that we're testing are having cytotoxic effects or not on non-specific cells. And we can directly compare immunotherapies like the BiTEs and the inhibitory antibodies and the CAR-Ts against each other and see how they're affecting stromal cells or other cells that are around the malignant cells.
And I would like to take that point to highlight maybe the advantage of CAR-T cell therapy over other immunotherapies, because a CAR-T cell is not a fixed immunotherapy. It can be engineered to become much more and to enhance its efficacy. We can potentially armor it against some of the inhibitory effects of this inflammatory microenvironment, or make it secrete a therapeutic payload or a cytokine to further enhance the potency and the immunotherapeutic effect. So we're very excited because I think calreticulin, as we discussed, is a fascinating target. It is possible that some of the other therapeutic approaches would work, but we think that maybe our CAR-T would be more suited. They're able to overcome even the worst cases of myelofibrosis, where there's a lot of inflammation, a lot of fibrosis, and more simple approaches may fail to tackle this challenging environment.
CAR-Ts are a very promising field. We've been really excited by this collaboration between the University of Oxford and UCL, combining the expertise of both of our laboratories. So it's been a really exciting project.
It has been a great collaboration. Again, Martin Hewley lab, which is the host lab, and Claire Roddy's lab provide the CAR-T cell expertise, and the CELLAC lab has huge experience in working with MPN and developing the organoid model. So we're very excited, and hopefully we will be able to move into a phase 1 trial where all the unanswered questions about safety and efficacy can be tackled, and then hopefully provide an additional, another hopefully curative treatment option for many patients.
That's all the expert insights we have for you today. It appears that the development of mutant CALR targeting strategies marks a potential new chapter in the management of MPNs, and we look forward to updating you on further developments in this area as they occur. Thank you for tuning into this episode, and make sure to check out the rest of our exclusive coverage of EHA 2025 on VJHemOnc.com. Don't forget to follow us on X at VJHemOnc to stay in the loop, and subscribe to our podcast on Spotify, Apple, or Podbean so you never miss an episode. Until next time.